This poster is entitled Whole Genome Sequencing to Detect Small Deletions in Ribosomal Protein Genes in Diamond-Blackfan Anemia Patients. DBA is a rare disease affecting roughly 1 in 5 to 10 million live births, where patients cannot make red blood cells. It is autosomal dominant with incomplete penetrance, and about 65% of patients it can be linked to mutations in genes encoding ribosomal subunits. Looking at Figure 1, it can be seen that 35% of patients with the clinical diagnosis of DBA, however, have no known mutation. Here we wanted to detect and investigate small deletions in ribosomal protein genes present in DBA patients without a molecular diagnosis. As shown in Figure 2, we conducted whole genome sequencing on 5 DBA patients without a molecular diagnosis. The bioinformatics methods highlighted in Figure 3 were used to identify larger structural variants ranging from hundreds to thousands of base pairs, as well as single nucleotide polymorphisms. We detected around 40,000 uncommon deletions and insertions in 5 different patients. 55 variants could be directly mapped to ribosomal protein genes, and 16 of those were unique to single patients or a small subset of patients and are presented in Table 1. Looking at Figure 4, RPL34AS1 is presented as an example showing the correlation of a 3200 base pair heterozygous deletion with 2 small heterozygous and 1 homozygous deletion, which in fact represents a common variation at the observed site. Due to the inherent imperfect nature of modeling and variant prediction, each of the predicted variant sites has to be experimentally validated in patient DNA. For this purpose, we designed flanking primers as shown in the panel on the upper right and did PCR validations generating the gel images shown in Figure 5. In case of the deletion in the intron of RPL27, shown at the top of Figure 5, 2 bands were expected and also observed, a 900 base pair band for the wild type and a 500 base pair band with the deletion. When submitted to sequencing, a junction site can be observed where the wild-type sequence starts to overlap with the sequence adjacent to the deletion, validating the existence of said deletion. While for both the intronic insertion in RPL26L1 and the exonic insertion in RPL14, the resolution of the gel is too low to allow to discriminate between the bands, sequencing shows the existence of junction sites where the insertions can be observed against the wild-type. In conclusion, we showed that deletions and insertions can be detected for DBA patients without a molecular diagnosis and validated for their causality using primer-directed PCR. We have detected 16 such variants in 5 different patients and are conducting the validation at the moment.